Good day, everyone. Let's discuss the physiology of flowering today. As we all know, a plant grows through different stages before it finally produces flowers and fruits and complete its life cycle. So, starting from the seeds, growing through germination, becoming a seedling, and then undergoing vegetative growth. Later, completing the vegetative growth, shifting onto the reproductive phase, starting to flower, setting of roots, and finally, production of seeds. These are the different developmental phases which comprises the life cycle of a particular plant. So today we will be discussing about the major or one of the most significant phases in the growth cycle of plant, that is flowering and reproduction. So, how exactly does plants know when to stop its vegetative growth and to shift to the flowering stage? Or how are the plants able to sense that now it is the time to go into flowering? Or are there any signals that tell the plants that now they have to stop the vegetative growth and shift to the reproductive stage and start flowering? So, if there are any signals such like that, then how are these signals being perceived by plants to enter into the reproductive phase, starting with flowering? So, let's check out some of the major points that lead to the flowering of a particular plant. So, what is flowering? Flowering is a transitional phase in the life cycle of a plant wherein it will be completing its vegetative growth or maybe stopping its vegetative growth for the time period and then shifting on to the reproductive phase, starting with the production of flowers. We all know plants produce flowers depending on what type of life cycle it is having. For example, a perennial plant will be producing flowers at a particular season, while an annual plant will be producing flowers within a short period of time because they have to complete the life cycle within a year. So, for perennial plants, the flowering can happen after maybe around 7 to 8 years after germination. So, depending on what type of life cycle the plant is following, the transitional phase from the vegetative growth to the reproductive growth via the formation of flowers can happen at different times in different plants. So there is immense importance for perpetuation and origin of variability, which can be reflected in the next generation, and this takes place by the transformation mainly of the vegetative apex into a reproductive structure. That is what is happening during flowering. During flowering, the vegetative apex gets transformed into a reproductive structure, in this case, flowers. And the change from the vegetative state to the reproductive state is termed as flowering. So, what are the major signals that tells a plant that it is now time to flower? So, in order for flowers to develop, there are mainly two important changes associated with it. First is the change from the vegetative nature to the floral state, and second is the commitment that it should form a flower. That means the signals has to be perceived and the signals will indicate that it's time to flower. So what could be these signals? It could be the maturity of the plant, or it could be certain environmental factors like the availability of photoperiodolite and temperature. So the regulation of vegetative to the reproductive transition may happen due to various factors or a combination of factors. These factors could be either internal factors or the external factors. Internal factors could include the different types of growth hormones that are present within the plant body, and external factors could be mainly the light and the temperature. So whatever be the signals that promote or inhibit flowering, these are found to be moving from the roots, cotyledons, or leaves to the shoot apex, where it has to effect the change of the vegetative structure into the floral structures. So once a signal is received by the meristem, then the plant will determine how to respond to that particular signal, and if all the conditions are appropriate, there will be a shift from the vegetative structure to the floral structure, thereby affecting flowering. So, as I said earlier, flowering evocation or the formation of flowers uh, can occur in the shoot apex when it specifically commits the apical meristem to produce flowers, and these could be due to two major factors, the internal factors and the external factors. Internal factors could be the shift in the change in phase, that is from vegetative to the reproductive phase, and as I said earlier, the hormones that are present within. The major external factors that would act as a stimuli for evocation of flowers include light, temperature, the type of light or the length, radiation, and of course, availability of water, moisture. So let us first check out how light influences flowering. There is a phenomenon which is known as photoperiodism, and this phenomenon is found to be very closely related to how plants respond to flowering. So let's check that out. So, what exactly is photoperiodism? Photoperiodism can be simply stated as the response by an organism to synchronize its body with changes in day length. What does it mean by? It means there is a biological measurement of the length of day and night that is being exposed to the plants, or how a plant responds to the relative length of day and night, and this responsiveness is termed as photoperiodism. And photoperiod is the duration of availability of light for growth and development, that is photoperiod. So at high latitudes, this responsiveness or the relative length of day and night is very important because it indicates the change in season. That means if a day is getting shorter and shorter, that means winter is approaching. So when the days get longer and longer, we could understand that summer is round the corner. So some plants regulate their flowering this way by sensing the change in the length of day or the night. So by sensing the difference of the relative length of day and night, plants regulate their flowering. And this responsiveness to the relative length of day and night is known as photoperiodism. So the whole phenomenon was put across by Garner and Allard when they were studying about tobacco, uh, certain variety of tobacco, wherein they could sense that the change in length could affect flowering in that particular variety of tobacco. And this has led to the understanding of photoperiodism in the first place. And the relative duration of light and dark towards the control of flowering in certain crops, that came to be known as photoperiodism. So, based on the responsiveness of the plants to the availability of light, that means the relative length of day and night, or simply to put, how they respond to photoperiodism, plants can be classified into three groups. One is the short day plant, second is the long day plant, and third is a day neutral plant. So we can have three groups of plants depending on the responsiveness to photoperiodism. They are short day plant, the long day plant, and the day neutral plant. In short day plant, The plant can only flower under day length shorter than its critical uh, day length of 24-hour cycle. So that means, so if there is a cycle of 24 hours a day, how much length it is needed for a particular plant to flower? So in short day plants, the length should be of the day. That means the availability of light should be for a shorter period, and then the flower goes into flowering. I mean, the plant goes into flowering. For example, in crops like strawberries, poinsettias, and chrysanthemums, the critical day length to induce flowering must be less than some maximum. While in the long day plants, it is just the opposite scenario where the plant will undergo flowering when the day length is longer than its critical day length of the 24-hour cycle, and the cabbage, beet, etc., all come under the this group of long day plants. Here, the critical day length must be longer than a minimum. So that is in the case of long day plants. Then what about the day neutral plants? Here, the length of light, that means the day or night, has got no particular effect in the flowering of this group of plants, and if all the other conditions are okay, they will flower anytime. Examples of day neutral plants include tomatoes, cucumber, eggplant, and bean. So, after they have been bred for a long time, most crops become insensitive to day length. For example, in rice and in some soybeans and in cotton, they have now shifted on to what we call as a day neutral plants. So, in short day plants, it needs a day length which is shorter than its critical day length. Long day plant needs a day length longer than its critical day length, while the day neutral plants are not affected by the length of the day or the night. So, what is this critical day period? It is a duration of the photo period or the dark period that ultimately determines whether the plant has to go through vegetative growth or whether it is now ready to produce the flowers. And this duration of the photo period is known as a critical day period. So this is a table which shows a photoperiodic response of some common plants, and you could have some of the examples like chrysanthemum, cosmos, dahlias, morning glory, rice, soybeans, tobacco. They all come under the short day types, while long day plants include barley, cabbage, henbane, onion, poppy, spinach, wheat. They are all long day plants. There are so many day neutral plants which does not respond to, which does not have much, does not get influenced by the relative length of day and night. For example, maize, potato, tobacco, tomato, chilies, these are all examples of day neutral plants. So let's take a look at the flowering behavior in short day plants and how the change in the relative length of day and night, that means the light and dark, are influencing the flowering behavior in short day plants. As I said earlier, short day plant needs shorter daylight and a longer darkness period for undergoing flowering. So that is the optimum that it needs, or the most conducive condition that is needed for the short day plant to go into flowering is shorter daylight and a longer period of darkness. So what will happen if the daylight increases and the period of darkness is reduced? This short day plants will continue with its vegetative growth and it will not flower. So, what happens if the light period is lesser and the darkness is longer? As we said, it should naturally go into flowering. But if at all, in any case, this long dark period is broken by flashes of light, then the plant will not get the signal to produce flowering. Instead, even if the light period is shorter, the plant will go into vegetative growth because the darkness has been broken by flashes of light. So, strictly speaking, a short day plant can go into flowering only when it has shorter daylight and an uninterrupted longer dark period. So, the uninterruptedness of the dark period is very important for the short day plant to go into flowering. Even a small flash of light that breaks the darkness could give the plant a signal that it should go into vegetative growth, and that flash of light could inhibit flowering in short day plants. So, how does a long day plant respond to the relative day length and the night? Flowering in long day plants is affected if the light period is longer than the critical photoperiod and it has got a shorter darkness. With lower light periods and a longer dark period, the plant will continue with its vegetative growth. Suppose this longer dark period is broken by flashes of light, that will give again a signal to the long day plant asking it to go into flowering. So, in contrast to how a short day plant will respond to the breaking of the darkness period, here in long day plants, even that short breakage or flash of light, which is breaking the long dark period, could also induce flowering in long day plants. That means, even if the light period is shorter, if the longer dark period is broken by flashes of light, the long day plant can go into flowering. And so, this is how the short day plant and the long day plant behave based on the relative availability of light and the period of darkness. So, this phenomenon where the darkness is being broken up by flashes of light has been explained as the night break phenomenon. For plants with a critical night length, a short flash of light in the middle of the night would make the plant behave as if it has been exposed to a long day. So, what does this mean by? Let's take some examples and see if by manipulating the length of night we could induce flowering. So, in short day plants, as you can see here in the example, it needs a shorter period of day length to go into flowering, and it needs an uninterrupted uh, darkness, hour of darkness to continue into flowering. So that is the ideal uh, photo period that is needed for the short day plant to grow into flowering. So suppose the length of day is increased and the darkness period is reduced. What happens? Short day plants, as you can see here, it will not go into flowering. But again, suppose the dark period is interrupted towards the end of the darkness, even then that short flash of light could inhibit flowering in the short day plants. Suppose the darkness period is interrupted in between, the same effect is being sensed by the short day plant, wherein the flowering is inhibited. In case of the long days, but we can give this plant or shift the plant into a dark place, thereby increasing the duration of dark period, we could induce flowering in short day plants. So, in a nutshell, what we should understand is that for a short day plant to go into flowering, it needs shorter daylight and a longer uninterrupted dark phase. If this is the case with short day plants, how could we manipulate the length of night to induce flowering in long day plants? As we know, long day plants needs longer light period and shorter night period for going into flowering. So that is the normal case with the long day plants. In case of the longer night period being interrupted, whether at the beginning, the end, or in between the long dark hours, if it is getting interrupted with flashes of light, these flashes of light are being sensed by the long day plants as signals to go into flowering. So even if the day length is shorter, the flashes of light that is appearing during the long night hours can be sensed by the long day plants as signals to go into flowering, and the plants will go into flowering. So this is what we call as a night break phenomenon, and how we could manipulate the length of night to induce flowering in short day plants as well as the long day plants. So this was all about the duration of light and how the photoperiod will affect the flowering in plants. So taking into consideration, we know that sun's light has got many colors. Okay, so does color of light influence the growth in plants, or maybe even flowering in plants? We all know sunlight gives off a lot of color of light, but during long day period, that means in long days of summer, the predominant color that we see in the UV radiation is blue. And we know that plants that were dormant in winter will start growing in spring. Seeds start to germinate. So what does that mean by? At those times we have blue light more, and seeds germinate, stem starts to grow, leaf starts to expand. That means blue light is needed for the growth of stem, stalk, and the leaves. The germinating seeds, seedlings, and transplants also need blue light. But what happens when the days are shortened? Maybe during the end of summer, the blue light that dominated the spectrum of sunlight is now slowly taken over by the light in the red band. And it is during this time that we see most of the fruits getting ripened, vegetables are getting ripened, and we are going into harvest during the late summer or even in the early autumn. That means red light is needed for flowering and production of fruit. So there is a connection between the color of light, the growth pattern, the flowering, and the production of fruits. So how the plant can sense the change in color and respond accordingly. There is something that is known as the phytochromes that are present in the plants, which helps the plant to sense these changes of colors. And as you know, phyto means something to deal with light, and chrome is something standing for pigments. So here, phytochrome is a pigment found in some plant cells that have been proven to control the plant development, and the pigment that controls growth and flowering in many plants. This pigment is known to exist in two different phases, which is one is known as a phytochrome red light sensitive form, and the second is known as a phytochrome far red light sensitive form. Phytochrome red light sensitive form is denoted as PR, and PFR is phytochrome far red light sensitive form. Let's check out what this is and how these phytochromes influence flowering in plants. The photoperiod mechanism we just saw, the phytochrome exists in two versions, and these two versions are interconvertible. In the sense, as you can see here, this is a phytochrome red sensitive, and this is a phytochrome far red sensitive. Phytochrome red sensitive form can absorb red light and convert itself into phytochrome far red light sensitive phase. And this phytochrome, far red light sensitive form, can absorb light in the far red region and get itself converted into the PR form. That means they are interconvertible. So depending on what is the length of light that is available, they can be seen as PR or PFR. If it is PR, it can readily absorb red light and get converted into the PFR form. The PFR form can readily absorb far red light of the spectrum and convert itself into the PR form. So how does this influence the flowering in short day plants and the long day plants? Let's check out. So how the whole thing is controlling flowering. So when there is change in day length, the photoperiod mechanism gets activated in the leaves as we saw earlier, and this activation of the photoperiod will lead to the activation of certain hormones within the plant which are found to be stimulating flowering. These floral hormones are termed as florigen, which is actually a hypothetical flowering hormone and which has never been isolated. But still, its presence is felt and its effect is seen when it produces flower buds and later the plant goes into flowering. So, when there is change in day length, photoperiod mechanism starts acting, which stimulates the production of a certain hypothetical floral hormone known as florigen, which initiates flower bud formation and the plant goes into flowering. In short day plants, what happens is the PFR is seen in abundance in darkness, because as we know, short day plants needs lesser light and longer uninterrupted darkness. So what happens? It will start absorbing the far red light, and then gradually the amount of PR, that is a phytochrome R, builds up, and the short day plants, the florigen starts getting activated. That is a hypothetical hormone which is influencing or activating the flowering, and the plant goes into flowering. So PFR is abundant in short day plants. It starts taking up far red light, then it gets converted into the PR form. Florigen is getting activated, and it ultimately results in flowering in short day plants. So what happens in long day plants? Normally, it is the PR that is abundant in it, and it starts taking up red light, which is abundant in the sunlight, and gradually the PFR starts building up. So once the PFR starts building up, the florigen is activated in the plant, and the florigen takes the plant forward to flowering. So, in a nutshell, as I said earlier, the phytochrome, either it is in the PR form or in the PFR form, they are interconvertible, and depending on the availability of light and what type of light is available, it can either be seen as a PR form, that is a phytochrome red light sensitive form, or the PFR form, that is the phytochrome far red light sensitive form. In a short day plant, the the PFR gets converted to PR, florigen is activated, it goes into flowering. In long day plants, it is the other way around. The PR gets converted into the PFR form, the florigen is activated, and ultimately the plant goes into flowering. So, this is how photoperiodism and the nature of light influences flowering via the hypothetical hormone, that is florigen. So, when we compare between the short day plants and the long day plants and how they behave to photoperiodism, we could see that short day plants flower when they are exposed to day lengths shorter than a certain critical minimum, while the long day plants are going into flowering when they are exposed to a day length which is longer than a certain critical minimum. Short day plants do not flower if dark period is interrupted even by a small flash of light, while the long day plants can be stimulated to flowering by breaking the long dark periods with flashes of light. In short day plants, normally they flower in early spring or autumn, while long day plants normally flower in spring or in early summer. Short day plants cannot be stimulated to go into flowering by treating it with gibberellic acid, while the long day plants can be treated with gibberellic acid, which could ultimately induce flowering in them. Short day plants needs a continuous dark period. That means it needs a long night period for flowering. So they can also be called as long night plants. Long day plants needs continuous light period and a short darkness period. So they can also be called as short night plants. So this is the difference between the short day plants and the long day plants and how they are getting influenced by the change in photoperiod, and that means photoperiodism, and how they flower based on the relative availability of light and darkness. Now let's check out another major factor which can influence flowering. Other than the light, temperature has got a major role to play as far as flowering is concerned in plants. So requirement of plants to go through a period of cold before they can flower, that is termed as vernalization. That means low temperatures can influence flowering in certain plants, and this cold period before they set to flower is known as vernalization. Clippart in 1857 was the first to notice that low temperature could work uh, with certain kind of wheat like winter wheat and spring wheat and could make them flower. While Lysenko was the first to use the term vernalization for low temperature treatment to promote flowering in plants, and the definition for vernalization as acquisition or acceleration of the ability to flower by chilling treatment was put forward by Choward in 1960. So with this, we could understand that vernalization is an important adaptation of plants growing at high latitudes or altitudes to ensure that flowering occurs in warmer days of spring or summer. That means it will allow sufficient time for the seed to develop before the onset of winter. So vernalization is a cold temperature treatment or a period which is needed for the plant to go into flowering. And this again is very important as far as agriculture is concerned because various crop plants, including certain winter cereals, need a prolonged period of low temperatures to initiate flowering in the spring. So without a period of low temperature, these plants continue with their vegetative growth and they fail to produce flowers and seeds. So vernalization is an adaptation to certain plants to go into flowering to ensure that seeds are developed before the onset of winter. It is an aerobic process, that means it requires oxygen and it requires metabolic energy. That means in the absence of oxygen, cold treatment will not be effective in inducing flowering. And this process also needs the presence of water because, as we know, seeds do not germinate in the absence of water. So, vernalization to be effective, we need to give them oxygen as well as sufficient amount of moisture. So how does this vernalization act? There are two major theories put forward to explain the mechanism of vernalization. One is known as the phasic development theory, and the second one is known as the hormonal theory. Let's check them out. Phasic development theory was first proposed by Lysenko in 1934, and according to the theory, there is a series of phases in the development of a plant, starting from the simple seeds to the germination, the seedling, the vegetative growth, the flowering, and finally the seed set. So each phase in the plant growth and development is stimulated by an environmental factor, which could be either internal or external. External, especially temperature and light, is very important, as we saw already. Then commencement of one phase will take place only after the completion of the preceding phase. And there are two major phases to the phasic development theory, that is the thermophase and the photophase. Thermo standing for something to do with temperature, and the photo standing for the effect of light on the growth and development. Thermophase depends on the temperature, and here we are discussing about how the low temperature can influence flowering and growth. Vernalization, as I told earlier, it is seen to accelerate the thermophase, and this thermophase should soon be followed by a photophase, which normally requires the presence of light. Then the second theory to explain the mechanism of vernalization was put forward by Melcher, who proposed that chilling treatment induces the formation of a new floral hormone within the plant system, and he named that hormone as vernalin. As we saw earlier in case of photoperiodism, where the presence of light is influencing the formation of a hypothetical hormone florigen, in vernalization, the chilling treatment is known to induce the formation of a type of hormone, again a hypothetical hormone, which is known as vernalin. And here, this hormone that is called as vernalin can be produced at one part of the plant and is easily transmitted to the other parts of the plant. And this phenomenon has been explained well or even proved by grafting a vernalized plant to an unvernalized plant. That means chilling treatment can affect the formation of vernalin, which can get transmitted from one part to the other. And this was proved by grafting a vernalized plant with an unvernalized plant and noticing that the unvernalized plant, after getting grafted with a vernalized plant, sets or initiates flowering. So the hormone vernalin is known to diffuse from the vernalized plant to the unvernalized plant, thereby inducing flowering. So here you can see how the hypothesis of hormonal involvement is explaining the mechanism of vernalization. According to Purvis, there is a formation of a substance known as A from some kind of a precursor, and this A will get converted into another molecule known as B after the chilling treatment. This B compound is very much unstable and it gets converted into the compound D, and this compound is what we have been calling as vernalin, the hormone which can induce flowering. So this vernalin becomes a precursor and it gets converted into florigen. We have seen what florigen is. It is a hypothetical flowering hormone. So once the florigen is formed, this can get translocated to the meristematic tips, where the vegetative tip is now getting converted into the floral form. That means it induces flower formation. So here, a precursor molecule forms a compound known as A. A, upon chilling treatment, gets converted into an unstable compound known as B. B gets converted into D, which is vernalin. Vernalin induces a formation of florigen, which is a flowering hormone, which get translocated into the meristem tips, which gets converted itself into flowers. So if chilling treatment can induce the formation of florigen, what will happen at higher temperatures? So the process of vernalization could be reverted by a high temperature treatment. That means when the temperature raises beyond a certain limit, that is in high temperature, this the unstable compound B, instead of getting converted into D-vernalin, it gets converted into another compound which is known as C, and this process of reverting is known as de-vernalization. So, to explain the technique of vernalization, the seeds are first soaked in water. Water is needed for the seeds to germinate. So it will germinate at a temperature of around 10 to 12 degrees centigrade. Then the seeds are transferred to low temperature treatment for a few days, spanning up to 30 to 50 days at a low temperature of 3 degree to 5 degrees centigrade. Then once after the chilling treatment, the seeds are allowed to germinate, and the plants uh, which grow up from the vernalized seeds are seen to go into flowering quickly when compared to the untreated control plants. So this is in a nutshell what happens after vernalization. The plants grow into quick flowering. So devernalization is a reverse process of vernalization, wherein by high temperature treatment, you can revert the effect of vernalization. That means devernalization is effected by treating the vernalized seeds or buds with a high temperature, and Lang et al. Demonstrated that application of gibberellins can replace the cold treatment for vernalization in certain biennial plants. So that means even in the absence of chilling treatment, by treating with gibberellins, we can have the effect of vernalization in certain plants, and the plants can grow into flowering. So, what exactly is a practical application of vernalization or the chilling treatment? Due to vernalization, the vegetative period of the plant is cut short, resulting in early flowering. It can increase the resistance of plants to fungal diseases. It increases the cold resistance in plants, again, which is needed for the winter sturdy plants. In biennials, vernalization induces early flowering and early fruit set. Flowering can be induced by grafting vernalized plant with an unvernalized plant, thereby making the process of flowering easier. And this is widely used in horticulture to make plants go into flowering easily. And definitely, this also helps in crop improvement because early flowering means early seed set. So, these are some of the practical applications of vernalization, wherein the chilling treatment can speed up the process of flowering and seed set. There are certain similarities between these two phenomena which influences flowering, that is photoperiodism, which is responsiveness to the relative length of day and night, and vernalization, which is the low temperature treatment. In both photoperiodism and vernalization, there are certain physiological properties or activities that is taking place within the plant that influence flowering in plants, and both these processes are mediated through plant hormones. We have seen florigen and vanillin, and both can occur naturally or can be manipulated. That means we can induce artificially, and they both have agricultural applications. So these are some of the similarities between photoperiodism and vernalization. As much as they have similarities, they do have the differences. The major difference between photoperiodism and vernalization is that photoperiodism is the induction of flowering in plants by exposing them to appropriate photoperiods, that is, the light and the dark periods, while vernalization is a process of induction of flowering in plants by exposing them to low temperature treatment or chilling treatment. Photoperiodism provides both the stimuli as well as induction of flowering, because sensing of light and dark itself can act as a stimulus which asks the plant to go into flowering, while vernalization only prepares the plant to perceive the signals which can lead to flowering. In photoperiodism, the stimuli induced by light is received only by the green leaves and it is not getting transmitted to the other parts of the plant body, while the stimuli induced by the cold treatment is received by the leaves, but it can move on or get transmitted to different parts of the plant, as we saw earlier. Photoperiodism is mediated through the hypothetical hormone called florigen, while vernalization mediates the hypothetical hormone vernalin, which is known to induce the synthesis of florigen in plants, which can ultimately take the plant into flowering. Exposure of 2 to 3 appropriate photoperiods is enough to induce flowering in plants, while exposure to low temperature for about 30 to 50 days is needed to induce flowering through vernalization. In photoperiodic induction, you cannot nullify the effect of exposure to light or darkness, that means unfavorable photoperiod, while the effect of chilling treatment can be reverted by exposing the plants to high temperatures, that is devernalization. Gibberellic acid has got no special uh, activity as far as photoperiodism is concerned, but it can affect or influence the flowering only in the long day plants, while GA can replace cold treatment to induce vernalization in all types of plants and they can take it to flowering. So, this is all about photoperiodism and vernalization. These are two major physiological activities that are influencing the flowering and fruit set in plants. Photoperiodism is the responsiveness of the plants to the relative length of day and night, and based on which we have three different groups of plants: short day plants, long day plants, and the day neutral plants. While vernalization is all about temperature and how temperature influences flowering, it is the chilling treatment which prepares the plant to go into early flowering. That is what is vernalization all about. So, thank you so much for your patient listening.